Hello student, good afternoon. Today we will start the topic gastrulation. Langston described in 1875 and Herbert in 1906 pointed out that the gastrulation is a process during which the single layered blastula is converted into two layered, example amphioxus, or three layered, most vertebrates gastrula. According to Balansky 1976, the process of gastrulation is one of the displacement of parts of blastodome of an early embryo. As a result, the endodermal and mesodermal organ rudiments are removed from the surface of the blastula and brought into the interior of the embryo, where the respective organs are found in differentiated animal. Thus, the single layer of cells, the blastoderm, gives rise to three germ layers: ectoderm, endoderm, and mesoderm. This definition embodies the concept of migration of specific organ forming areas. In other words, the gastrulation may be defined as the dynamic process during which the major presumptive organ forming areas of blastula become arranged and recognized in a way which permits their ready conversion into the body plan of the particular species. Major events of gastrulation. Gastrulation involves the following events. Number one, arrangement of blastomeres of the embryo by means of morphogenetic movements. The three primary germ layer gets differentiated. Three primary germ layers get differentiated. Second number, the rhythm of cellular division. That is the cleavage is slowed down of almost inhibited. Third number, growth, if any, is insignificant. Fourth number, the type of metabolism changes and the rate of oxidation is intensified. Fifth number, the nuclei become more active in controlling the activities of the embryonic cells. The influence of paternal chromosomes of the embryo becomes evident. Sixth number, during gastrulation, chemodifferentiation is started by the synthesis of new and different kinds of proteins. Seventh number, the three primary germ layers are differentiated. Three primary germ layers are the differentiated. Morphogenetic movements. Next is the morphogenetic movements. Gastrulation is essentially a process of movements of parts of the embryo. Each part remains in the position into which it has been brought by the preceding movements. As a result of these movements, new structural elements are created in gastrula such as archenteron, notochord, neural tube in place of the simple layer of cells found in the preceding stage, blastula. The movements have created new shapes. Movements have created new shapes and new forms. Therefore, they have been referred to as morphogenetic movements. Walk in 1925, Trankons in 1969, the morphogenetic movements that occurs during embryogenesis are of irreversible nature, that is, each part remains in the position into which it was brought by the preceding movements, while those of adult are of a reversible nature. The morphogenetic movements appear to be movements of large part of whole embryo which stretch, folds, contracts or expand. Each moving part consists of numerous cells, and the movement of the whole would be an integrated result of the movements of the individual cells. Thus, morphogenetic movements are not under the control of the embryo as a whole, but are due to the integrated results of the movements of individual cells. During gastrulation, some specific type of morphogenetic movements of blastomeres occurs. In the majority of cases, two or often more of take place together. These are grouped under two heads. Number one, epibolic morphogenetic movements or epibolic. Embolic morphogenetic movements or embolic. Both of the words, namely epiboli and emboli, have Greek origin and they mean two different kinds of motions. The word emboli means to throw in or thrust in. The word emboli means to throw in or thrust in, while the word epiboli, on the other hand, denotes a throwing on or extending upon. Thus, these words through have opposite meaning but occurs simultaneously during gastrulation. Number one, epiboli or epibolic morphogenetic movements. Epiboli or epibolic morphogenetic movements. Epiboli is the spreading of prospective ectodermal cells, micromeres, over the inwardly moving presumptive notochordal, mesodermal, and endodermal elements of the embryo in holoblastic eggs of amphibians and cephalochordates. The micromeres of the presumptive ectoderm present in the animal hemisphere divide in rapid succession and produce large number of cells which migrate downwards and spread over the macromeres of the presumptive endoderm. In flattened blastula of reptiles, birds, and telosts, the epibolic movement are represented by antero-posterior extension and expansion of blastomeres associated with the peripheral migration during the formation of primitive streak and the separation of the germ layer. Second number, first is epiboly, second number is emboly or emboly morphogenetic movements. The embolic morphogenetic movements are concerned with inwards migration of prospective chorda, mesodermal and endodermal blastomeres from the external surface of the blastula and their extension along antero-posterior axis of the developing embryo. This inwards movement of the cells is due to innate factors arise from within a particular cell group or are depend upon the movements of other cell groups. According to Belinsky in 1970, blastomeres move inwards because these cells form an epithelium, the blastoderm, which is distinctly polarized. The distal surface of the blastoderm is in contact with external environment, while its proximal surface faces the inner milieu of the embryo. The inwards morphogenetic migration of prospective chorda, mesodermal and endodermal blastomeres in different groups of chordates may occur by the following methods. Number 1, invagination. According to Oak, the term invagination means insinking of the egg surface followed by the forward migration, which involves the displacement of inner materials. The process of gastrulation by invagination occurs in relatively unmodified conditions in primitive chordates like branchiostoma. In most higher vertebrates, this process is greatly modified and usually accompanies other methods. The accomplishment of gastrulation by invagination may be explained as follows. Let us consider the case of a Cceloblastula having a central blastocele and surrounded by the blastomeres. The upper hemisphere of the blastula may be regarded as the animal pole and the lower hemisphere as the vegetal pole if the vegetal half of the blastula is pushed in or invaginated till it touches the animals half lying opposite to it. The hollow sphere becomes transformed into a two-layered gastrula. During this transformation, the blastocele becomes practically obliterated and a new cavity termed archinteron has made its appearance. The cells of the animal hemisphere now constitute the epiblast, and original vegetal cells lying beneath the epiblast now constitute the hypoblast. The newly formed opening to the exterior constitute the blastopore, and the rim of the edge of the opening is called as lip of the blastopore or germ ring. Okay, thank you.